Good morning, everyone. Welcome to the webinar. I'm Joko Logis from BMG LabTech, the microplate reader company. Today, we have a very special guest, which is Ms. Shalini Markandan from Forest Research Institute of Malaysia. The topic today that we will discuss is ORAC assay. So we'll give a bit of an introduction in terms of ORAC, as well as discussing the relevance for the industry, as well as the academics. In terms of format, today we'll try to do something a little bit different. As you can see visually here, we are not actually in a forest, but be rest assured we are in a safe space following the SOP here. So Shalini will start with a 5 minutes or so of presentation describing ORAC in general, and then after that we'll straight away go to the question and answer session. So in that question and answer session, I will act more as a host, where I'll ask a series of prepared questions that I feel personally are quite important for Shalini to address. And then in the middle of that question and answer session, if any of you in the audience have any question, feel free to send them via the chat function in Microsoft Teams. Now, before I let Shalini start with her presentation, I will introduce Shalini a little bit in terms of her background, in case you're not familiar with Shalini before. So Shalini serves as an assistant researcher at the Natural Products Division, Forest Research Institute of Malaysia. She has more than a decade of scientific research and technical experience in handling ORAC, testing for the industry as well as the academics. Her main area of expertise is in the natural products, antioxidants, as well as anti-aging research. Shalini has optimized more than 10 bioassays related to this area, and she has also been involved directly in ISO 17025 accreditation of ORAC assay in Forest Research Institute of Malaysia as technical manager and SAMM approved signatory. So if there's one person that you can ask more about ORAC, Shalini is definitely the right person. So we are very lucky to have her here today. So Shalini, do you want to start with the presentation?
Yes. Okay. Hi, Joko, and hello, everyone. Thank you for joining us, and thank you for the brief introduction on myself, Joko. And it's a great pleasure to be here today to discuss about this exciting topic, which is the ORAC assay, industrial and academic relevance. Okay, let's move on to the presentation. Firstly, what is ORAC? ORAC stands for Oxygen Radical Absorbance Capacity. This is an in vitro analytical method which is developed by the scientists of National Institute of Health and also USDA, and it was also later commercialized by the Brunswick Laboratory. So this ORAC assay, we use them to quantify antioxidants in foods, botanicals, ingredients, and also the biological samples. And ORAC assay is very popular among the research and also food industries. Okay, moving on. Okay, what does ORAC measure? Actually, ORAC measures the antioxidant capacity of a substance against ROS. ROS is reactive oxygen species. There are few types of ORAC assay that is available. Okay, first is the ORAC hydrophilic, which this assay actually measures the water-soluble antioxidants. Next is the ORAC L, also known as ORAC lipophilic. It measures the lipid-soluble antioxidant capacity. And soon what happened that the ORAC assay has been improved and designed by the Brunswick Laboratory. They designed it as ORAC MRO, ORAC multiple radical, also known as ORAC 6.0. Yeah, it actually measures the sum of the antioxidant capacity against 6 different reactive oxygen species, namely the peroxyl radical, hydroxyl radical, singlet oxygen, superoxide anion, peroxide nitrate, which is from the reactive nitrogen species, and also hypochlorite, which is from the reactive chlorine species. Okay, next I'm going to tell you how ORAC works. ORAC applies a simple principle here where it actually measures the fluorescent intensity and also the degree of reactive oxygen species damage. And please take note that the fluorescent actually is the fluorescent probe. Yeah, when the radicals is initiated, okay, in this case we use AAPH to generate the peroxyl radical. So then the radical will oxidize the fluorescent. In other words, they will attack or kill the fluorescence. What happened then? The fluorescent will be decomposed, they will be decayed, and the signal curve will actually go through reduction. So you can see the graph on your right hand side. You can see there is a curve without antioxidant. You can see that the curve has dropped. Yeah, in the event there is antioxidant present, the radicals actually are neutralized, so the fluorescent is preserved, and they are also protected from the free radical damage, and there will be no reduction or less reduction of the fluorescent signal. So you can look at the graph that the curve which is labeled as with antioxidant, they are extended longer. Yeah, and the highlighted area in the graph area, okay, is the net area under the curve, which is used to calculate the ORAC value based on the Trolloc standard calibration curve. So that's all about the ORAC principle, Joko.
Okay, I think to simplify it to maybe those of you who maybe never heard of ORAC before, I think so if we have a sample that has a high antioxidant capacity, then you will see that the fluorescent signal goes down later, so it will last longer. If the sample has a lower antioxidant capacity, then the signal goes down faster.
Yes.
So this difference is what we are measuring.
Yes, exactly.
And we are using Trollocs as a standard,
yes,
as an antioxidant standard. All right, I think that concludes a very short presentation. So I think we will straight away go to the meat of this discussion, which is the question and answer. So again, if any of you have any questions that you would like to ask, please feel free to send them via the chat function. So the first question that I'll ask is that we know that there are many different types of ORAC assays, sorry, not ORAC assays, but antioxidants assays. So how does ORAC differ? So why do people do ORAC? What's so special about ORAC?
Alright, actually ORAC is a very unique assay, I would say, because in general they are very stable because it's using the fluorescent, which is excellent photostability. So the assay is actually very stable, and ORAC is reproducible, they are cheap, and they are also actually designed in an automated for high throughput screening for the 96 well format and also for the 384 well format, and also it's adaptable for various type of samples as well, example like for juice, extract, and also biological samples. In terms of their mechanism, it measures the radical chain breaking ability of the antioxidant, especially the secondary antioxidant. It also allows the measurement of a slow and also the fast acting antioxidant as well. And additional to that, it actually combines the both free radical inhibition time and the degree of inhibition by antioxidant into one single quantity. It also uses the biological relevant radical sources, example like peroxyl radical.
Okay, I have a follow-up question here. So I think the interesting part that I catch here is the slow and fast acting antioxidant. So as far as we know, I think quite a lot of people know as well that most antioxidant assay, I think, are endpoint assays. So you only see one point of data basically. So doing ORAC basically allows us to have a more broader view, a kinetic view, to see maybe which of your sample act faster and which of your sample act slower, for example. Okay, so that's definitely interesting. So my next sort of topic that I would like to cover is definitely on the reason. I think in the last few years there has been some academic discussion, I think, that sort of warned against overstating the value of ORAC importance basically, or rather the importance of the ORAC value itself. So Shalini, in your view, is ORAC still a relevant assay for the academics as well as for the industry?
Alright, Joko, before touching on the relevance, okay, let me brief you about what are the critics that ORAC went through. Yeah, what actually happened is that the marketers, they mislead and they misuse the ORAC value for the marketing purposes, and ORAC has become some sort of marketing terms. It's like kind of a ORAC brand. So the marketers started to use ORAC value to rank the foods, sell their foods, and also to determine the choices of food. And they also use this ORAC value actually to explain the antioxidants to the public. Okay, so without any context, and people or the consumers, they will definitely believe that the ORAC acid and complete antioxidant, it is a perfect package of nutrition to them. Yeah, and they also make some claims that the high ORAC value means that a high antioxidant in the human body, without taking into consideration of the ADME, we call it, the absorption, distribution, metabolism, and also the excretion. Yeah, and other than that, some also compare the ORAC value of different ingredients. For example, like juice of an orange with the extract of a mango, for example. And some even they borrow the ORAC value of the other company for their own ingredient without testing its own own ingredient
Which
itself.
is a bit unethical as well.
Yeah. Yeah. So there are many debates on ORAC, which leads to the reason why the USDA has brought down or withdrawn the ORAC database from their website in year 2012. Okay, now I will explain to you why ORAC is still relevant or not. Okay, when I did some research, when I explored the Google trend, and in the Google search engine, surprisingly, world is still looking or finding for ORAC. Yeah, and the statistics shows that the trends of ORAC search in year 2000 to 2020 to 2021, as shown in the figure there, and there are a few companies that are still searching for ORAC in the net. Yeah, and besides that, there are also recent publications found in year 2021 related to ORAC assay, which I pull out few of them that I already put out there, so you can have a look. And moreover, FRIM is still receiving samples from the industry for ORAC testing without fail, and which I believe that the ORAC is still a useful, valuable tool despite its controversy. This is because it still remains as an interest, valid, and also reliable option for the preliminary evaluation and also for screening of antioxidants.
So as I understand it, well, when academics or researchers do ORAC assays, then they get the ORAC value. What they do is that they report it in a journal paper or journal article. So let's say if an industry do their own research, normally they wouldn't publish it, but to your knowledge, how do they use the ORAC value?
Okay, actually you can use ORAC values in few ways, and you can make them more meaningful if you use them in a proper way, So you can use ORAC value to discover. Yeah, you can use ORAC value to discover new sources of antioxidants, and also you can use ORAC value as a benchmark for the product development, which involve your standardization, optimization, processing step, and also extractions. And ORAC can help you to classify your products as the product with antioxidant property. And you can use ORAC value for the quality control purposes, which involve the storage, packaging, the stability. You still can use ORAC to measure. And another thing that you can use is to look into the rancidity studies, where you know that ORAC has a potential actually to prevent the rancidity by preventing the auto-oxidation and also lipid peroxidation in food. Yeah, and the most important thing is that it can be a precursor for further investigation at the cellular level and also in vivo level.
All right, thank you for that answer. I think quite a lot of us maybe have never done ORAC ourselves. So I think definitely there will be questions on what actually is the ORAC procedure or the general protocol of ORAC itself. So maybe Shalini, you can share a little bit on that as well.
Okay, this is a very general and quick protocol. Okay, firstly, you need to prepare a different dilution of Trolox. Second, you need to prepare the buffer, phosphate buffer at the pH 7.4. This will act as the medium for your ORAC assay. And then for the working wells, you need to have the standard sample and also buffer together with the fluorescence. And then you incubate your plate for 30 minutes at the 37 degrees Celsius. If you have a microplate reader with the thermostat features, you can incubate your plate in your microplate reader itself. And then once the incubation is complete, then you can proceed to the microplate reader. So make sure that your microplate reader has the fluorescent mode reading, yeah, and the plate will be read at excitation of 485 nanometer and also emission of 520 nanometer. Next will be the injection of the ROS. This can be done manually, and also you can do it automatically if you have an injector in your microplate reader. Then you have to proceed for the reading. For the assay to complete, it will take about 125 cycle, which each cycle is about 90 seconds. So you can see the curves at the figure B. So that is the complete experiment. And the calculation of the area under the curve is done by using the mass data analysis software. Yeah, so you can see the integration and the equation of the ORAC value as shown in the figure A. So it's quite a bit complicated actually. And plot the standard calibration curve of Trolox as shown in C. And then you can express your ORAC value as a Trolox equivalent. To be more specific, you can express them as a micromole Trolox equivalent per 100 gram.
Alright, thank you for that, Shalini. I will sort of go back to the earlier point that you made just now about different versions of ORAC assay. So there are ORAC assays that handles hydrophilic samples, then there are ORAC assays that handles lipophilic samples. So in your experience, well, I assume that handling lipophilic samples sounds a bit more difficult.
Yeah.
So is that true? How does that differ actually?
Yeah, in terms of their sample preparation, definitely it differs because you need a sample, you need a special lipid solubilizer to extract out the lipid soluble antioxidants prior to the testing.
Okay, so that's more like a pre-ORAC step.
Yes.
Or prep step in a sense. Okay. Then my next question is in terms of, let's say we follow all this procedure and then we got an ORAC value. The ORAC value by itself, without any context, won't have any meaning, right?
Yes.
So how do we report the ORAC value effectively?
Okay, this is the most crucial part, I would say, and this is my humble request as well to people all out there. Please make sure that the ORAC the ORAC analysis is done the ORAC analysis is done to compare the similar products. For example, you have an extract of a particular plant or anything, so make sure you use the same species of plant to do your comparison. And please make sure that the analysis is done in a single and also qualified lab, and also please make sure that the analysis is done using the same method. Yeah, and for the reporting of value, please make sure that we report using the equal amount of product. For example, like Trolox equivalent per 100 gram basis.
Okay, so when you say that analyzing using the same method, that means it must be the exact same protocol.
Yes, exact same protocol.
And if we are talking about a single qualified lab, means if you send to lab A,
yeah,
then if you want to compare, make sure you send to lab
the same
A again.
lab. Okay. Because we are not sure whether maybe lab A or lab B might be using a slightly different or modified protocol.
Oh, all right. Okay. So if we go back to the procedure, in your experience, for first-time ORAC assay runner, what do you think is the main challenges in terms of doing ORAC itself?
Okay, the challenges or the hurdles that we went through when optimizing ORAC assay is that the data analysis, because as I mentioned that there is 125 cycles. So when we are doing the assay in a high throughput format, so mean that usually you use a 96 well plate and you need to calculate the 125 cycles for 96 wells. So it's total about like a 12,000 area under the curve that you need to calculate manually. Yeah.
So that's definitely quite a challenge. So imagine if, let's say, just now you mentioned 12,000 data points. So calculating 12,000 data points using Excel, for example, that would be quite tedious, I would imagine. So I'll definitely have like a demo to show the audience here. So as Shalini mentioned, her calculation actually used Mars data analysis software by BMG LabTech. So this software only comes with the BMG LabTech reader. But before I show a demo of that software, I'll just like to touch on one more question since we're at the procedure. In terms of the injection, just now you mentioned that if the microplate reader has a automated injector that it can actually do the automated injection of the radicals itself. So you don't have to manually pipette it to the
Yes.
to the plate. So obviously we all assume that automated injection will be better, but how much better it is actually? Do you think the
Okay.
results will differ that much?
Okay, because this is based on my own experience when I've done both ways, automatically and also manual pipetting. So definitely there will be some changes, in terms of their measurement. And when you use the automatic injector, so meaning that they will give you more consistency in terms of the measurements.
Okay, that's good to hear. Okay, so now what I'm going to do is I'm going to do a demo for you, so you can have a look at our software and how we process the data. So this is a look of our software, the data analysis software. This is an ORAC assay. So here you can have some blanks in triplicates and some standards. Here we have 6 standards and some unknown samples, which is the one we presume to have antioxidants. And then here, normally people will look at the raw data. So here we'll have a raw data. Okay, we have the raw data for blank, standards, and all the others. This is at cycle 1, and we can go to look at the data from the last cycle as well. Okay, so there are many ways we can visualize this data, but the most interesting way, of course, is to show the signal curves or how the curves progresses over time. So let's look at just the blank and the standards. So just let's take that. So here we see the progress of the curves. Okay, so the standards that have higher concentration obviously have better antioxidant capacity. It will go down further.
Yeah.
So we are trying to measure the area under the curve. So all the one that goes down further will have a bigger area under the curve.
Exactly.
So it will have a bigger value, so to speak. We can also do all sorts of things, blank correction, average, because we have triplicates and blanks here, and we can apply all this data there. So the next thing that I'm going to show is the standard curve creation. So all these are already done automatically, so you just need to tick to show the curve. Here we have quite a nice standard curve based on the Trollope standard that we use, and these are the fit results. So using that standard curve, basically, we can automatically calculate the final calculated data that we want, which is the ORAC value, using this method. Basically, just tick that, and then you can see the concentration of the unknown samples here. Alright, so basically the way this software handles all this data is basically by using a template. So basically when we use a template, you can actually customize calculations for many different type of assays. So if you're doing MTT or DPPH assay or whatever other assays, we can actually help to automate the calculation for you, so you don't have to go to Excel. Having said that, if you want to export all this data in Excel, we can also do that as well. Okay, so that's it about the demo. Maybe to ask impression from a customer, because Frim actually has 2 of our microplate readers. So the first one is the Flowstar Omega, who has been there for 10 years, I think, and is still running. And recently you also have the Clarostar Plus. So the Clarostar Plus also are used for the ORAC assay. So in your opinion, what do you personally like or maybe dislike about BMG LabTech microplate readers?
Okay. For the BMG LabTech microplate reader, the features that I really like that is it has the high sensitivity, because we are running our ORAC assay in a very small volume, so we definitely need a microplate reader with a high sensitivity. And then next is the built-in injector. Actually, it gives me opportunity to simultaneously inject and also to read the measurement. So that's the very good thing, and it comes with the predefined ORAC template as well, which make my work very easy, fast, and also simple. And the interesting thing that I like is that I can observe the real-time progress, where actually the kinetic reaction,
This is the slow and fast acting thing
yeah,
that
so
we
we can
talked about
see
just now.
the kinetic reaction, especially after the injection of the free radicals, and definitely it's easy to use with just a simple click.
Okay, that's
Yeah.
very nice to hear. So here you can see a picture of me with Shalini. I think this was last year, I believe, still in the middle of the pandemic. We are still wearing masks then, and we are still wearing masks now. Although the situation has improved quite a lot, I think. So the next question, or rather sharing that I will request from Shalini is that I know that Frame itself, your lab also accept external samples, as you mentioned. So in terms of the range of services that Frame has, is ORAC included in one of the tests that you can offer, and what are the range of services that you can
Okay.
provide?
Yep. Okay. Definitely. You can find ORAC in Frame under Natural Product Division. So just to talk about the ORAC assay, the ORAC assay in Frame we optimize in year 2011 to 2013. We focus on the hydrophilic ORAC, which is the water soluble, and our ORAC assay is actually accredited with ISO 17025. Under the scheme Akreditasi Makmal Malaysia in year 2015 until now, and the samples that we are accepting for services are the plant extracts, vegetable, herbs, fruits, and also spice derived products, which includes the herbal products like tablets, capsules, powders, herbal drink, fruit juice, health foods, food supplements, and also tea products, including the plant extracts. Yeah, and other technical services that is also available beside ORAC and also antioxidant assays, which is the biotherapeutic evaluation, which includes the anti-inflammatory, cytotoxicity, anti-diabetic. We have all these services. And other than that, we do have phytochemical analysis and also plant identification services as well. So for the full list of services that available in Frame, you can just go to the link provided there, or you can just scan the QR code to download the whole list of services that available in Frame. So to be specific, under Natural Product Division, I think you can go to the page 27 onwards, if I'm not mistaken. Yeah.
Okay, so definitely looking at the samples that are accepted for testing, there's like a huge range of samples. So if you, for example, if you have any problem handling your own oral assays because your samples may be lipophilic or whatever, you can definitely contact Frame to consult, and also maybe perhaps send the samples for them to test. So thank you for that. So I have two more questions left from my side. So far we haven't received any questions, which can be a good or a bad thing. But if you have any questions, please feel free to send the questions, because after I finish my last two questions, we will straight away move to the next part of the today's program, which is the quiz. So my next question will be on the future of ORAC. So how do you see the ORAC assay itself develop into the future?
Yes, okay, ORAC in the future. This is my hope, yeah. So I hope that the industry will recognize the importance of understanding the strength and also the limitation of ORAC, okay, and the use of ORAC maybe can become more standardized in future. And another thing is to complement the ORAC values with the biorelevant studies, for example, like the bioaccessibility, bioabsorption, and also bioavailability studies.
So that's quite interesting. I think maybe you can expand a little bit about these biorelevant studies. What sort of assays are they? Are they like a different version of ORAC assays?
No, actually, it's a maybe can be a complete represent the physiological functions. For example, maybe you can go for the digestion processes. So from there you can see whether they are also penetrating into the cells or not. So for example, like the cellular based antioxidant studies and things like that. So you can see whether they are really penetrating into your cells or not. So that's the most important thing.
Okay, so these are like those CAA or
Yep.
CAP-E assays.
Yes.
Okay, because I think it's quite important, again, like Shalini mentioned, because the ORAC assay that we do is in vitro. Technically it's on a test tube or a microplate. So again, this does not necessarily correlate to the actual health effects that's in the body. But moving on the journey towards affirming that, we can do some in vivo studies to see whether the antioxidants are actually absorbed by the cell or not. I think that's quite important as well. Okay, so my last question will be in terms of the your concluding thoughts. So, in your conclusion, any important message that you want to convey to the audience this morning?
Yes, definitely. As the conclusion here, that I wish to stress the important foundational facts here, where the ORAC is definitely a valid and a useful representation of antioxidant capacity, and the ORAC is not the conclusion for the whole antioxidant story or the whole nutritional story, and no in vitro assays that can quantify the in vivo outcome.
Okay.
Yeah.
Okay. Finally, we have one question, very exciting. Question from Michael.
Yeah.
May I know for lipophilic samples,
Yeah.
normally whether it's better to use acetone or 50% acetone for extraction? Or may I know which type of solvent is more recommended? Thank you. So that's on lipophilic question. I expected there will be questions on that. Yeah.
Yes. Okay. For answering his question, you can use randomly methylated cyclodextrin, which is a very good. We call the lipid solubilizer, or we call it lipid enhancer for the extraction of lipid based antioxidants.
Okay. So I hope that answers your questions. All right. So before we move to the next part of I think maybe the second most interesting part of the program, which is the quiz, where you can win some prizes. Okay, before I do that, few more questions pop up.
Oh yes, sure, no problem.
Okay, compared to the NADPPH test, I think this is DPPH test, I believe. Is ORAC more sensitive or easy?
Yes, ORAC is more sensitive assay compared to DPPH.
Yeah, I think to
In terms
yeah.
of easy, maybe in terms of handling ORAC, I mean DPPH assay is more simple compared to ORAC, because in terms of time and for the preparation, ORAC definitely will take more time compared to DPPH assay. But in terms of their sensitivity and accuracy, I would recommend ORAC.
In terms of the microplate reader technology, as I'm the representing BMG today, I think I can explain a little bit as well. So if we are doing ORAC, that is a fluorescence-based method. If you are doing DPPH, that's a absorbance-based method. Comparing any similar kind of assays for sync function, if you can choose a fluorescence method and if you can choose an absorbance method, choosing a fluorescence method will always be more sensitive.
Yes.
That will be the fact, because absorbance, there are many things that can affect the reading. For example, if you have impure samples, if you have contaminants or clumps or whatever, it can actually affect your reading, as you can see. Whereas if you do fluorescence, for example, in this case, the fluorescence signal will directly correlate with the strength of the antioxidants itself.
Yes.
So even though it might have some impurities here and there, it doesn't really affect the interaction that much compared to absorbance. One more question. Sorry. So just now that question is from Sari C. So thank you again for that question. I hope that answers helps. The last question I have is from Dr. Assistant Professor Dr. Bhaskaran Gunasekaran. How long it will take for analysis to be done if we send samples to frame? Very important question.
Usually it takes about 14 working days to complete the
14, so that's around 2 weeks plus.
2 weeks plus, yeah.
Okay, all right. Okay, so if there are no more questions, so I'll show you, before we move to quiz, I'll show you a little bit of marketing video. I hope you don't mind. So we'll show you a product video for Clarastar Plus. So Clarastar Plus is a microplate reader from BMG LabTech, and the special thing is that it has the multi-mode capabilities, so it can read fluorescence, absorbance, and luminescence, and many more as well. And the other special thing, the second special thing, is that the Clarastar Plus has the LVF, or linear variable filter monochromator. So this is completely different from the traditional monochromator that you have in a traditional microplate reader, because this LVF monochromator is as flexible as a traditional monochromator, but at the same time it has the sensitivity that is almost similar to filters, because it can give you a higher signal. Okay, so this is again a completely redesign of a traditional monochromator, and it's our own patented BMG LabTech technology. So have a look at the video.
The new Clariostar Plus is a high-performance multi-mode microplate reader that fits the current and future needs of your laboratory. Flexibility combined with the highest sensitivity in its class makes the Clariostar Plus the most versatile instrument for all laboratory applications. Predicting the signal intensity of experimental samples is difficult, especially in kinetic tests. Simplify your assay setup with enhanced dynamic range technology, where both high and low intensity signals can be acquired in a single run. For almost any test, users can read a plate without any optimization steps. EDR uses the best settings for every plate. The Clariostar Plus, with its patented LVF monochromator and tunable dichroic mirror, provides the sensitivity of filters without compromising flexibility. Adjusting the bandpass down to 8 nanometers allows high-resolution spectral scanning, while broadening the bandpass up to 100 nanometers provides increased sensitivity. In absorbance tests, you can instantly collect a full high-resolution spectrum with a dedicated spectrometer. The Clariostar Plus has excellent sensitivity in luminescence, breadth, and alpha screen assays. Data quality in these assays can be affected by microplate designs, which allow light from neighboring wells to cross over between one another. Through crosstalk correction, apertures are automatically used to reduce noise from adjacent microplate wells, thus improving the signal-to-blink ratio. For cell-based assays, the Clariostar Plus with atmospheric control enables oxygen and CO2 regulation, as well as gas rampings. Hypoxic conditions can be maintained at oxygen concentrations as low as 0.1%. The Clariostar Plus can include both a red-sensitive PMT and a low-noise PMT. In this configuration, far-red fluorescence measurements are possible while maintaining high sensitivity in luminescence and alpha screen tests. BMG LabTech offers a unique and intuitive data analysis software, MARS, that allows users to easily perform a variety of data reductions and complex mathematical calculations critical to life science research. The new Clariostar Plus is the most versatile instrument for all laboratory applications. Contact BMG LabTech today at bmglabtech.com to arrange a demonstration for your lab. BMG LabTech, the microplate reader company.
Alright, I hope that video is useful and interesting for you. So as you can see, if you're dealing with cells as well, if you're growing cells, we can actually put atmospheric control unit. So if you're growing any anaerobic bacteria or anaerobic cells, then we can actually control the gases inside the cells, and you can just put the cells on a plate, leave them on, and then just go away and get your results later. So we have a few more questions here, so I'll just go to the questions first. Dr. Bhaskaran again, there's another question. So he says that one antioxidant assay itself cannot be conclusive in terms of the values, I think in terms of like concluding the antioxidant capacity.
Yes.
So based on your experience, what other assays can we use to complement with ORAC assay?
Okay, to complement ORAC assay, you can use the CAA assay, which is the cellular antioxidant assay. Okay, which you can, some of the assays, they use the erythrocytes, so you can also use that type of assay to complement the ORAC assay.
Okay. The next question I have is from Michael. If say we are dealing with a 96 samples, so I think this is 96 raw samples, may I know does the speed of addition of the free radical initiated solution affect the readings if we were to manually pipette to all wells? I think we sort of touched on this just now. Yeah. So again, this is I think a question on the manual pipetting compared to automatic injection.
Yes, definitely there will be a difference, because when you introduce your free radical into the 96 well, so when you start on your first well, by the time you finish your 96 well, the reaction already start occurring. So definitely it will affect your readings.
So I think like Shalini shows in the protocol earlier, so definitely she used our reader, and our reader has an automated injector on board. So you can actually use the automated injector. Basically you just have a vessel or tube with the radicals, and then it will automatically inject at the cycle that you want for all the plates. So the advantage definitely is time, because asking the machine to inject for you will be way faster than using our hand, for example. Yes. So definitely there will be some signal difference, I would say. So I think before we go to quiz, I'll just give you show you our contacts first. So in case you have any further questions, so you can contact us later. Again, I'm Joko from BMG Lab Tech, in case you forgot. So that's my email there. So I'm basically the technical support for the APAC region, including Malaysia. So you have any questions on applications, or you're asking for a demo, for example, you can contact us as well. If you want a direct contact in Malaysia, you can contact our exclusive Malaysian distributor, which is the Projin Links Sdn Berhad. There's a Mr. Lims is a contact person there. There's an email and a phone number. Now, if you want to send your samples, like one of the questions just now, I believe, then you can contact directly the director of the Natural Products Division at FRIM, which is Dr. Ling Swee Kiong. Her phone number is there and the email is there. So I'll just leave this slide on for a few more seconds before we move to quiz.